So today I'm going to be talking about antigen presenting cells. So antigen presenting cells are cells that present antigens on their surface using MHC1 or MHC2, which I will talk about in a second. They will then present these antigens to either your CD4 T cells or your CD8 T cells, depending on the situation, and they will cultivate some sort of immune response within your body in reaction to these antigens. Antigen presenting cells consist of dendritic cells, macrophages, and B cells. It depends on either MHC1 or MHC2. MHC1 is really all nucleated cells except for red blood cells. MHC2 is really just specific for dendritic and macrophages and B cells. So what is MHC? When we talk about MHC, we're talking about the major histocompatibility complex. So these are cell surface proteins that will display antigens to your T cells. Think of them as billboards. They pick up antigens and they show them on the surface of a cell, and then the T cells, like I said, will then cultivate some sort of immune response. For MHC1 specifically, these are displaying antigens to your CD8 cells, so your cytotoxic, your killer T cells within your body. And these are specific for antigens that are being made within a cell. If your body is picking up any sort of activity within a cell, your MHC1 is responsible for presenting those antigens to your CD8 cells. The way this actually works is that there's a structure in your cytoplasm, your nucleus, called proteasome, which will break down a polypeptide chain, a big protein, whatever you want to call it, and it'll divide it up into singular peptides that are very small in order for them to be transported into the endoplasmic reticulum via TAP. So TAP is kind of like a transport structure that will transport these peptides into the endoplasmic reticulum. Once inside the endoplasmic reticulum, they will bind to MHC1. MHC1 will get packaged up into a membrane-bound vesicle. It'll push through the Golgi apparatus out onto your cell surface, and then once on your cell surface, it'll present them to CD8 cells. CD8 cells will look at them. They'll be like, okay, this is what we're targeting, and they will go attack those cells. So MHC1 is pretty important just for targeting infected cells, cells that are already infected, and viral cells. It's also good at preventing natural killer cell activation. You may be wondering why that's important, why natural killer cell activation is negative in some ways. The overall picture, or in the grand scheme of things, natural killer cells are constantly checking your body, you're constantly checking your cells and looking for MHC1 activity. They're making sure that MHC1 is being expressed. If cells are not expressing MHC1, or if there's a low expression or low activity, they will look at these cells and they will automatically assume that these cells are infected, cancerous, whatever, and they will start to, of course, attack these cells. Now, this could obviously lead to some autoimmune complications within your body. So as long as MHC1 is being expressed within your body, you are avoiding those complications. Next up, we have MHC2. So MHC2 is specific for CD4 cells and is specific for external antigens within your body outside of the cell. The cell will essentially eat that antigen. It'll go to an endosome, which I drew out here. It'll go to an endosome and it will meet up with the MHC2. Now, MHC2 is already blocked by something. There's already something bound to it called CLIP, which is just another peptide. There's another protein called HLA-DM, I believe is what it's called. HLA-DM will facilitate the transportation or the switching of CLIP and the antigen. And then, similar to MHC1, it'll get bound up and it'll travel to the surface of your cell and it will present them to CD4 T helper cells. And then the T helper cells will they will talk to whatever cells they need to talk to target these antigens, right? So as we can see, CD4 cells are your helper cells, CD8 cells are your killer cells. So if MHC1 is the billboard presenting antigens that are being made within a cell, that would be MHC1. And then MHC2 would be the billboard that is presenting antigens that have been found outside of the cell externally, and antigens that have been eaten up by your cells, if that makes any sense. So I hope I made this super easy. I know it's a lot of material. It's definitely a lot of raw material when I was learning it, but I hope I made this simple to understand. And yeah.